Hi students, in this lab we are going to be going over stem cells, and I'm sure you've heard a lot about them in popular culture and debates and different news things that have come out. And in this lab we're going to explore those a little bit deeper and learn how they are used, and you are even going to practice seeing what it's like to replicate stem cells. So in this first part, what is a stem cell? You are going to do some background reading on what stem cells are, and using this reading here, you are going to answer the questions that are down below, like, how are stem cells stem cells are unspecialized? What does that mean? How do they self-renew, and how do they differentiate? Remember that Science Takeout is really good at always putting the answers to those questions up here. So make sure you're very thorough with those answers. And then you're going to continue on part 1. And you are going to look at this reading here with stem cells in the lab and understand more. Please do this reading really thoroughly, highlight it, write on it, whatever you need to do to understand it and remember it. But this section breaks out what the difference are between adult stem cells, because adults, like you and I, we do have stem cells in our body, what they can do and their limitations, and then embryonic stem cells, how they are used and what they are capable of. Okay, and then make sure that you understand here these diagrams below and how they relate to that reading. When you are done with that, you are going to answer these questions, but you're going to use the information on the page before that you read in order to do that. Make sure that your answers are very thorough there. Okay, then you are going to do some more reading on stem cells, and then you are also going to do this reading in particular is very, very important. So make sure you go through and highlight it very carefully. But then it's going to ask you to take the development diagram sheet that is provided, and you are going to use the reading from here to put these different phases of stem cell research in order. Okay, so you're going to use these and you're going to put them in order, and you're going to make sure to record the correct order, and each of the letters is listed up here about and you're going to figure out what comes first and second, etc. And then you're going to use that other information that's here to answer the questions, question 5 there. So then we are going to head into part 3 and understand how stem cells can be used to treat different diseases. In this part, you will start doing some of the testing. So make sure that you read this in the background information so that you can proceed on to the next part. Also, since this is the testing phase, it's important to note that Science Takeout always has safety guidelines and what you should do as far as safety instructions about not eating or drinking anything that's here. Make sure you have a clean workspace, that it's uncluttered, good lighting, etc. So make sure you read over those safety guidelines before you start your testing. And we are going to flip over to the testing part. Please go through this and highlight page 5. And then when you are finished with 5, move over to 6. On page 6, you are going to be using this diagram here, this drawing that they have here, to fill in this data table that's here. So when you're looking at this diagram here called Growth Factors and Stem Cell Differentiation Flowchart, you are going to look at how they have a stem cell here, and then using different growth factors, they are able to change the cell, and then they add another growth factor, and then they get an outcome here. Okay, and I'm being intentionally vague here because I want you to figure out the answers here. But essentially, you're going to see that here it says muscle cells used to treat heart damage What growth factor do you add first to get a muscle cell? So you're going to look, and where it says heart muscle cell or a nerve cell or pancreas cell, which one of these is a muscle cell? And whichever one is a muscle cell, you're going to work backwards from there, and you're going to look, what was the first growth factor that was added? What was the cell outcome? So the first growth factor you're going to write here. Then the second growth factor that's added, you're going to write here. And then, the first growth factor, you're going to write here. The second growth factor, you're going to write here. And then you will get a color in your, because you're actually going to do this experiment. You're going to get a color in your test tube after you add the correct growth factors that are labeled here. When you add the correct growth factors, you will get either a red or a green or a blue solution, which represents the heart muscle, the nerve muscle, or the pancreas cells. And I meant to say nerve cells there, but you are going to get those three different ones, and they're three different colors, so you're going to write the color there. So you're going to use this chart to fill out this data table. You will not get the answers to column 4 until you do something later. So first fill it out for muscle, the 2 growth factors and what color you got, the nerve, what 2 growth factors and the color you got, and then the pancreas cell, the 2 growth factors and the color that you got. From there, you're going to retire this diagram for now, and then you are going to read down and start doing the actual test. Now, I like to have my chemicals all lined up. Here's my clean cups that it will ask you for. They are these tiny clean cups that are included. And then I always like to have my my pipettes next to my test tube, so you can see this is growth factor 1. I like to have it next to growth factor 1, and growth factor 2 I like to have next to growth factor 2, and I have it all lined up like that so I can grab it really cleanly. And then over here is embryonic stem cells, and here is adult human stem cells. Again, all of this is just simulated things, but it's all a simulation for this lab, but it's all there. So you're going to follow the instructions. It says use the embryonic stem cells, ESC, the dropper from that, which is here, my embryonic stem cell, ESC, dropper to add 10 drops of the embryonic cell culture to a small clean cup. So I'm going to take this, I'm going to take out 10 drops. I'm never going to cross-contaminate, so I'm never going to use this pipette on one of these other tubes. I'm just keeping this pipette for this tube. And then I'm going to put 10 into a cup, and then it says add 1 drop. When I close this all up and keep it together, I'm going to close that up, and it says add 1 drop of the first growth factor you selected. So you have to go back up to the table and get a growth factor that you selected, and add 1 drop of the first growth factor. So if I picked this first one, like growth factor A, then I'm going to put 1 drop of growth factor A using its pipette into the same tube. And then I'm going to put growth factor 1. If I'm trying to create this muscle cell, I'll do growth factor 1 in there. And then it's going to ask me, if you succeeded, you should be able to turn it into a certain color. And the color here was red for muscle, so this should turn red. And if it did succeed in turning red, I'm going to write yes there. And if not, I put no. So I'm going to do that same thing in number 3, in a new clean tube, and number 4. And then I'm going to write the answers to those questions, okay? And then I am going to flip over to the second part of this. And now I'm using adult stem cells to treat disease. So the first one we were using, I want to show you the difference. We were using embryonic stem cells, okay? This next time, we're going to be using adult stem cells to treat a disease. So we're going to be using these ASC, the adult stem cells, in our cup. So we'll need 10 drops of that in each one. But you are going to go down and you're going to fill out this chart the same way off of this like diagram drawing. And you're going to fill out the chart. But remember that column 4 is going to happen after our testing. And then you're going to flip over to page 9. You're going to read through, and this time using your adult stem cells and the adult stem cell pipette. You're going to put 10 drops of this in a cup, and then you're going to use the growth factor that you put on your data table because you wrote out the directions. You're going to put the growth factor and see if you got the correct color. And so you'll write yes if you did or no if you did not. Then you are going to summarize that here on number 4 and 5. And then you are going to flip over on the back to a little bit of a debate where it says, here's, why some people say, yes, we should be using stem cells for research and development. And some people say, no, absolutely not. It's a little bit of the debate there. And you write the pros and cons of that. And then this lab, make sure to upload a picture at some point of you doing the work. You could have these little test tubes full or halfway, or you can take a selfie doing this, but make sure you do that as part of the lab to get full credit. So that's how you complete this lab on stem cells from Science Takeout.